Okay, so welcome everybody. So we are very happy to have our research director here today, Dr. Ashley Wazana. And yeah, I'm relieved we have a really good turnout online. I saw that there's not many people in the amphitheater today, and that's probably because whoever was here 2 weeks ago when it was -20 and no heat, they they learned their lesson, are in a nice warm space today. So that's very smart, but it's actually better today. So yeah, we're really happy to have our research director with us. Dr. Wazana is a senior FRQS clinician scientist and associate professor of psychiatry at McGill University. He's director of the psychosocial aspects of disease research axis and director of the Early Childhood Disorders Psychiatric Day Hospital at the Jewish General Hospital, and director of the Dream Big Consortium, which is what will be the topic of his talk today. And he leads a bench to bedside developmental research program focused on how biological vulnerability and environmental adversity interact to shape mental health treatment response and resilience. So it's a great pleasure. We welcome Dr. Wazana.
Very great to see. Can we see the audience in here? I just want to view. Yeah, I want to see them here so that at the same time make sure that, yeah, that way when if anything comes up, there's in the view. There, no. Yeah, that we can put that on that side.
You want to put it on this side? You're going to see
on that side.
here,
It's fine.
maybe?
Okay, great.
Okay.
Thank you.
Well, when you're going to click, it's probably going to go away. Unless you...
It's fine.
Oh, it's
Yeah.
okay, works.
Okay. So thank you for joining. And yes, I think a few people learned the lesson. I think it's slightly warmer, but it's still. I'm hoping to be able to share today what is a part of the research program that I've been leading for a few years, and which is really a program. I'm just going to start with my conflicts of interest, which are none, but which is scaffolded on so many different people who I work with across a few countries. Everybody from my lab, David Laplante, Yifan Wang, and Ting Zhang, who are all, sort of capital and instrumental people inside the lab, to some of the people who've sort of started the study that I've been involved in from the beginning, Michael Meany and James Kennedy, and then Esther CK, who joined us with Celia Greenwood at McGill as well, who've been really instrumental in some of the biostatistics. Kristen Connor from DOHAD, who I've worked with in trying to understand the way that we can translate this into modifying sort of, risk assessments. And then there's a number of cohorts that this is just generalized to. There's the ALSPAC cohort in the UK with Anthony David and Jonathan Evans, Rebecca Pearson. There's Generation R in Holland with Tonya White, Henning Tiemeyer, and Ryan Mutzel. We also have Gusto, where Michael Meany is there, but also Tene Pang from Norway. We have Mona Beckes, who's at Oslo. There's also Jai Lati, who's in Finland, and Tim Oberlander, who's at UBC, and then Craig Olson, who's joined us with the ATP study from Melbourne recently. So this is just to say that this is an endeavor to work with existing data sets in a manner to sort of in a concerted fashion ask questions in a replicable manner. And so all of these people have been instrumental and really in the formulation of the hypothesis and the questions that I'm going to present today. My objectives is first to speak about a developmental model for psychopathology, and understand a method for using multiple data sets for replicated research, and then try and understand what are the cognitive, relational, social, emotional processes that actually make the link between the developmental factors and emerging psychopathology. And you'll see why that's important. The context is that mental disorders are lifelong. We're here in the Institute of Community Family Psychiatry with adults. We are just across the lane with child psychiatry, but 75% of the cases that appear here in adult psychiatry had symptoms that started before the age of 18, and 50% of them before the age of 14. Okay? And those with earlier manifestations have the most severe trajectories. But what is really the most important statistic I want you to retain from this whole talk is that there have been very little, if no change, if not an increase in the prevalence of mental disorders over time. Absolutely none. We've actually gone up. In contrast, there have been dramatic gains in other disorders. Dramatic gains. We have actually gone, in French they say, du surplace à la première place. We actually haven't really moved much. And now all the other ones which were leading the pack when it came to like morbidity and quality of life and, are now below us. So then we are now in first place. Okay. And the explanatory value of existing models doesn't even warrant an intervention to prevent. We just don't have enough information to say if we mobilize this at a larger scale, we could actually anticipate making a significant dent. And depression then, by design now constitutes the greatest cost to healthcare. Okay, what an interesting place to start, which is, why as a child psychiatrist, this is the work I do, is the developmental origins. Barker had hypothesized a few years ago, it's now actually a theorem, it's no longer a hypothesis, that many diseases originate from environmental experiences during pregnancy. Some of the work done looking at the impact of the blockus in Holland during the Second World War and the impact that had on the emergence of autism and schizophrenia later on was formulated into what we call a developmental origins of health and disease model. And prenatal stress in multiple ways can be measured. Depression, anxiety, pregnancy-specific stressors, which is different than these formal diagnoses, but just worries and preoccupations during pregnancy. And even social environmental adversity have all been seen to contribute to the emergence of mental disorders. Just a little link to the book on the right, which is the first book that I co-edited with CK and Tim Oberlander as a summary of the complex, but really real literature on the importance of prenatal stress on child development. And prenatal depression, which is the most common prenatal complication, occurs in 14 to 23% of women who struggle with some symptoms of depression during pregnancy. I think there might be, there was something about how we don't agree. We don't really know yet. Okay, I'll come back to that. There are actually mechanisms linking prenatal stress and development. Some of neurodevelopmental in terms of changes in brain regions that are responsible for stress and impulsivity and emotional regulation. There are changes in the HPA axis at level of the cortisol pathways. There are changes even in the placenta and the way that the placenta grows is vascularized and even permeable to stress hormones, and even epigenetic changes which actually modify profoundly the way that certain genes are expressed. All of them having an impact on the way glucocorticoids sort of travel across the placenta and influence early child development in utero. We know that prenatal stress is associated with increased fussiness, negative behavioral reactivity and fearfulness, increased internalizing and externalizing behaviors. Internalizing are more the anxiety, depression, sadness, avoidance, whereas externalizing are more opposition and conduct and hyperactivity. And this is an interesting study that was done by one of our colleagues in Finland, which examined mothers during pregnancy every 2 weeks between the 12th week and 40 week of pregnancy, and asked them about symptoms of depression, and then dichotomized the group into those that had clinical levels of depression and those below. And then as a function of their pregnancy reports of depressive symptoms, then mapped the outcome of internalizing symptoms, externalizing symptoms, and just overall symptoms, according to a measure called the Child Behavior Checklist administered between the ages of 3.5 to 5. And what you will see is consistently the two groups which is this group is the mothers who reported threshold symptoms of depression at 12 weeks, 14 weeks, 16 weeks, 18 weeks onwards, those who didn't. The two groups differ significantly in the amount of symptomatology at any time point across pregnancy, which is really one of the questions that we have always is are there specific windows, critical periods where exposure should be of greater concern? And what's also interesting that when these are three different types of analysis. The last one is adjusted, and it's adjusted for postnatal depression. And you can see that, of course, the association goes down between maternal depression symptoms during pregnancy and child psychopathology, but it doesn't go down to zero. So it is in part explained by continuing symptoms of depression, which we don't know which comes first. Is it, chicken or the egg? But either way, that in and of itself does not write off the entire effect of prenatal stress, okay, or prenatal depression in this case. Okay. We know that it's also associated with the increased rates of depression at age of 18. Each standard deviation increase in prenatal depression is associated with a 1.2 times increase in depression. And also it increases the rates of ADHD. And what's going to be interesting is the sex-specific effects of this. But there's little agreement on how one measures prenatal stress. There's so many different ways to do it, and that's going to be part of the focus of our work. There are individual differences to prenatal stress, meaning not every child responds the same way. These are twins. These are also twins. Over time, expressions, these are identical twins. It really speaks to the fact that environment matters, and that one wonderful sort of systematic review was done a few years ago by Abbott et al. That really tried to summarize the differential effect of stress on childhood outcomes as a function of genetic diversity. So it just speaks that genes do make a difference. And so any model really needs to consider genetic susceptibility. And this is a great study in 3 different samples, independent studies, United States, Italy, and Germany, by the lab of Daniel Weinberger, which really looked at the joint effect of genetic susceptibility to schizophrenia in something called a polygenic score. I'll speak about it a little bit more later, and the early life complications. And what was interesting is that the influence of those genes depended on early life complications. And we're talking about even having to do with pregnancy and all sorts of complications around pregnancy. And the genes identified that were sensitive to early life complications which were associated with schizophrenia were actually genes highly expressed in the placenta. So it gives us a clue as well of where some of these genes are actually operating, and that these genes were differentially upregulated in the placenta for the male fetus. So again, that there are sex-specific sensitivities to stress for different psychopathologies. And this is some of the work of Tracy Bale, and that's really looked at the impact, specifically mapping at the frontier of the uterus and specific genetic and epigenetic modifications of the placental expression of genes by male and female, and even parent of origin. So there's really a very nice sort of experimental work to document how this actually works at the interface of the placenta as well. So Michael Meany's work here at McGill shed light that the early environment modifies not only parenting behavior in offsprings, but also gene expression and neural connections behind these changes. And it did so in animal models of rats. And then there's work done by Jonathan Mill, which was one of the first labs along with ours to show that early maternal behavior then modifies these outcomes in humans. So it's not just about developmental susceptibility, it's also what the early environment can contribute to modify that. We're going to talk about that a little bit more. So the summary, if I want to summarize this, I want to speak that mental disorders are lifelong. We're going to start doing something about them. We have to start early. They constitute the highest cost to healthcare. The explanatory value of models is quite modest and needs to be larger. And many developmental factors need to be included in one model. And we then can look at causal and protective factors in these models as plausible targets of early identification and intervention. So our research approach is about a replicated research. The reason being is that a couple of studies I'm going to highlight really sort of bring this to the fore is that 1,500 scientists were polled in Nature in 2016. 70% of these scientists said that they had failed to reproduce at least one another's experiment. 50% had failed to reproduce one of their own experiments. This is another very important one, especially in field psychology. 270 researchers attempted to reproduce the results of 100 experiments that had been published in 3 prestigious psychology journals. This was published in Science in 2015. This succeeded only 39 times. And these are landmark studies that define the way we think about child development and adult development. So, there are multiple factors at play. Study designs can be confusing the results. Repeat testing, meaning you multiply examine a certain association until you can find something that might just be statistically sort of happenstance. Specific conditions in different environments where you're studying that have not been measured as cofactors. Sometimes the journals are not great. There's a lot of journals out there that rely on studies, and so their methods of review might not be as stringent and might not be asking some of the questions. I remember submitting a study and having important questions asked back and realizing, that I had made a mistake, and then we had to rerun some of the results as a result of that. And that's part of what the peer review process does when it's not necessitates or requires the funding of a journal. And there's downright falsification. But all of these are really probably in decreasing, frequency, of these factors. But these factors exist. So then the idea is that if research is going to be helpful and meaningful and for the purposes of modifying policy, you just need for it to be replicable. Okay. And so what we set out to do is really identify cohorts that are comparable. And if you can do that, and harmonize them, then you can start to ask the same questions in many studies at the same time without having to start new studies. And that's a very cost-effective way of managing science. So that's one of the first lines that we write in our grants for CIHR, that we're saving money because we're just really using other existing studies instead of having to create them. There are sets of harmonized measures that now are recommended for new studies, but those are really nice for new studies, but not for existing ones that have now already results into teens and adulthood. And so we can already start to ask the questions without waiting till I'm a grandparent or great-grandparent. So there's a study in Montreal and Hamilton called the MAVEN, which had about 590 studies, dyads. These are all prenatal cohorts that also looked at genetics, prenatal stress, maternal-child interactions, and a number of outcomes, social, emotional, and even psychopathology over the lifespan. There was one in UBC that we've included. There's one in England, in Bristol, which is the Avon longitudinal study, 15,000 dyads. There's one in Norway, as I said, that's 90,000 dyads with genes as well. So it's quite impressive. Prado has 4,500. There's one in Holland called Generation R, 97 dyads. Some of these have imaging as well. Half a dozen of these studies have imaging that we can use. Gusto is a study that was literally designed on a platform that we had created in Montreal, which is MAVEN, because Michael Meany was really the principal investigator for the psychopathology outcomes, and the Australian temperament study. So you can see that this has tried to breach, via various diverse regions of the world. And these studies have ongoing data collection. They're not done. They're really just, and you can see these are like very complicated slides to just tell you how many measures are being administered at what time points. And what I just want to just show is that they're all different and they're all different time points. Nobody sort of sat down and said, we're going to do this at this time point in this measure. Everybody has their fetish measures, their comfortable measures, their time points, and some of them are guided by financing. Anyways, if you get funding or not, you can miss a certain time point. So the point then is to work with these data sets and then try and harmonize them in a manner that makes them comparable. And we harmonized everything. We harmonized the exposure variables, and then we harmonized the outcome variables. That way, even if they did not ask the same questions, we can find something way that they're comparable that we can construct the same factor, like a latent factor. I'm going to talk about that. A big one when we're looking at the impact of early development is social environmental adversity, and that includes like life stress, contextual stress, personal stress, and interpersonal stress. And these are the questions, your partner died, you moved house, the results on the test suggested your baby might not be normal. So that's a life stress. And contextual, you became homeless, housing defects, your income was reduced. Personal stress could be early parenthood, maternal education, substance use. And interpersonal stress was, partner hurt you, you argued, you're divorced. And what's interesting is each study has some variant of some of these questions. They're not exactly the same, but what we do is we go look for them and construct similar scores, for life stress, for contextual stress, for personal stress, and for interpersonal stress. Gusto also has these questions, and so does Generation R. These are all different scales that they use, but ultimately we go look for the individual questions and try and create something which is similar, and we create a factor that has life events, contextual risk, personal, and interpersonal. And this gives you an idea of the fit statistic to see how comparable they are across the 3 different studies that we did it in. And we did the same thing for maternal mood. So we have the environment, and then we have the actual experience of symptoms during pregnancy by the mom. And again, everybody uses different measures, whether it's the Edinburgh study or the Crown CRISP or the CESD or the PSAS. There are a number of measures, but there's some questions that are about anxiety and depression, some about somatic symptoms, some of them are about pregnancy-specific worries. Because all of those are very specific domains. Each of them have their own thought to be their own contribution. So again, we sort of created a factor. This is general psychopathology, which is basically what is shared among all of these symptoms, and then what is specific and just residual for anxiety, depression, somatic, and pregnancy-specific worries. And we were able to again construct 3 factors in each of the studies that were very comparable in the fit. So it means that even if they had different studies, different measures, we extracted the questions which could be comparable. We had to do the same thing for genes. Some of you might be familiar with the GWAS, which is basically a large-scale examination of all the variants in the genome, and then we had to harmonize that genome, the GWAS, in a manner that you could construct a score, and that includes everything from like minor allele frequency, which ones you take or you don't take, what's the threshold before you don't use a certain allele, the quality of the imputation, and even the imputation pipeline. So a number of things you have to do to harmonize them, and then create a score, which is a sum of the contribution of the many variants in the genome. Each of them have a small effect in contributing to a specific phenotype, like ADHD, depression, anxiety. And that score is sort of summed all the individual contributions of each of these genetic variants. There are like 100,000 of them, and you just sort of can, depending on the threshold you pick of significance, you will add up, up to 100,000 SNPs to only 10 or 5 if you only take the ones that really have a high correlation. And you can find genetic susceptibility scores for depression, ADHD, multiple disorders. This is a Psychiatric Genetic Consortium, is an organization that tried to create them systematically. EAGLES was one that finally started to construct one for children. And these are the ones that we use. So you can see these are about 16 of the best ones. And then our research approach was then to harmonize also the outcomes, because we didn't want to only be able to compare exposure, we also wanted to compare the outcomes. And I'm going to speak about an approach for comparing outcomes that really reflects a sort of a trend now towards a different approach to nosology, which is called the hierarchical taxonomy of psychopathology. That's an effort from various mental disciplines to improve organization and description of measurement of psychopathology. So it tries to look at dimensional approach that cuts across traditional DSM. It tries to capture the variance that's common and then find what's specific and residual. The fact is there's so much overlap between diagnoses that there is actually core variance that could be extracted, and then you can find what becomes residual to like internalizing or externalizing. And then you can even use inputs from multiple informants, including children, parents, and teachers. and take symptoms across the spectrum. You don't have to have categorical. And these are valid in children and in adults, although in adults they start to look a little bit different over time. But this is to give you an idea of how many different measures, these studies use. They're all different. The Child Behavior Checklist, Strengths and Difficulties, the PAPA, the BPI, the Berkeley Puppet, the SCARED, which is a measure of anxiety. These are for externalizing. These are all the different measures as well that are being used. And even for the child, for generals, like we have all of these different measures. So you can take a measure and tag each of the scales to whether it, all of them belong to general psychopathology, all of them contribute to a general factor, but then they could also specifically contribute to internalizing or externalizing, a bit like this. So you can think this is probably oppositional problems, ADHD problems, rule breaking and aggressive. All of those contribute to externalizing, but they also, and just like depression, anxiety, and PTSD contributes to internalizing, all of them contribute to a general factor. This looks like some of the nail and string art from the 70s. You might have one of those in your garage still left over, but it gives you an idea of when you have as many subscales as we did, all of them would load on from all the different measures on the general factor, but some of them only on the external, some of them only on the internal. And these are the raters, the parent, the child, the teacher, and the child. So we usually sometimes have up to 3 raters. And we were able to create a factor, again, with diverse measures of psychopathology that was comparable so that we could then look at the same measure as an outcome and the same exposures across those studies. We did the same thing for ADHD, although it was complicated to get something that was specific for inattention and hyperactivity. But we created one for parents, one for teachers, and even a composite measure. And again, just to give you an idea that the parent measures and the teacher measures and the child measures are quite divergent across various studies. So that was what we need to do. And once we did that, then we start to build a model. And the model was, how does these developmental factors, these early influences predict psychopathology? Because then when we can start to have replicable associations, then we can start to, feel we have something that's certain that can be modified, and then we can hunt down the mechanisms. Okay. So that was the first study that we did. We took all 3 studies and compared mood symptoms during pregnancy and psychopathology between the ages of 4 and 6 in children in 3 different studies, and found that just general mood symptoms of the mom during pregnancy predicted general psychopathology symptoms between 4 to 8 years later, consistently across the 3 studies. We also looked at pregnancy-specific worries, which was very interesting because it's not just mood symptoms, but just specifically worries about pregnancy that was also contributing to psychopathology. You can see this is 0, that would be no effect, so it's just to the right of that. And when the line breaches this, it means that the confidence interval is slightly, it makes the result not as significant. But when you sort of sum up all 3 of those, which are in the same direction, you can see that it really strengthens the association that never breaches the 0 mark. And finally, we also looked at whether general mood factors predicted internalizing symptoms, so more anxiety and depression, which we know there's a very specific effect. And in the 3 studies consistently, you can see that there was also. So now we know in 3 studies that maternal prenatal mood symptoms, whether it's anxiety, depression, life-specific, pregnancy-specific worries, predict psychopathology 4 to 8 years later. Then we thought, okay, can we do the same thing for the genes? And so we looked at all these 16 genes and how do they predict general psychopathology, specific internalizing and internalizing. And again, there are like about 4 or 5 of them: cognitive ability, ADHD, neuroticism, schizophrenia, and insomnia, that were all predictive. So the genetic susceptibility for those predicted outcomes in the same cohort. So really the idea was, okay, we can see the stress matters, the genes matter. The Abbott study said genes and stress together, work together. So we need to do that in a replicated manner. And so it requires novel computational approaches because we're talking a lot of genetic information, a lot of adversity information, and doing that across multiple studies. So that's what we've been working on for the last 5 years. I have to say it's been more complicated than I thought, but because we also brought MOBA, which is a Norwegian study, which has 10,000 subjects out of the 90 or 1,000 or so that we could use in order to replicate this. Even if we had findings which were consistent within our 3 cohorts, we thought we wanted independent replication in another study that was absolutely set up differently in the modeling. And so we predicted general psychopathology, like I said, the sort of shared variance and the specific variance for internalizing and externalizing using general random forest, which is an approach which sort of tries to parcel down the data into various trees of divisions, of nodes, of disorders, and of genetic scores. So we fit that individual, each study we fit the association, we try and find what's the best cutoff, then we group them all together and we see what is the best cutoff for the genetics gene, the polygenic score. And then after that, we assemble all 3 together in the same model and adjust mutually for all the other ones. This was what we did for the 3 studies in Alzheimer's, maybe in the Generation R. This is what we're doing for the 4 subcohorts of MOBA, so that we can then have that replicate. Like it's a double replication. This is replicated here, and then it's replicated across another study. So in Dream Big, what you could see is A is social environmental adversity. So the impact, what are the genes that modify the influence of social environmental adversity on general psychopathology, internalizing and externalizing. That's A. And then you can see what are the genetic scores that modify the influence of maternal mood symptoms during pregnancy on general psychopathology, internalizing and externalizing. It's not consistent. Some of them affect more general psychopathology, some of them more affect internalizing symptoms, some of them affect more externalizing symptoms. So for example, ADHD, when exposed to social environmental adversity, increases general psychopathology and decreases internalizing disorders. And you can see that for prenatal mood symptoms, neuroticism, so the risk for having neurotic symptoms decreases when you have mood symptoms during pregnancy. It decreases the risk for having general psychopathology, but it increases the risk for having internalizing. So it just gives you an idea that the genetics susceptibility responds to different environment to predict different kinds of outcomes in mental disorders. We did this for ADHD. And in this case, we looked at the ADHD genetic susceptibility score, the PGS, and the 2 environments, both maternal mood and social environmental adversity, which we combined together to see how that predicted ADHD reported by teachers. And what's interesting to see here is that this is a 3 individual studies. That's a summary effect. But you can see that there's an effect for genes on ADHD. There's an effect for environment, which is social environmental adversity and maternal mood, which is prenatal stress. There's an effect for sex. So this is girls have a lower rate of ADHD. And there's also a sex-specific effect of adversity, so that we know that the effect of adversity is worse in males, even if just after adjusting for genetic susceptibility. So that was also we did the same study trying to predict brain regions that are associated with ADHD. And we tried to look at how prenatal stress and sex interact to predict some of these brain regions, how prenatal stress and the ADHD genes predict those changes in the brain regions, and the same, we tried to include all 3 of them. What was interesting is that across all the studies in brain development, we expect that there's a sort of a curvilinear relationship in terms of cortical growth and then trimming and pairing. And we know that ADHD is a maturational disorder, so there's a delay in trimming and pairing of some of the regions that are associated with ADHD. So we could see that was happening in both of the studies. Gusto, we have 4 to 6 years old, whereas Generation R, we have 10 to 14 years of age. So they're situated in different part of the actual curve. And what was interesting is for both of those studies, you could see the specific effects happening. So for the Gusto study, where you would expect some of the growth, there was a slowdown in the growth specific to having ADHD vulnerability and having more prenatal stress. Versus the generation R, which you expect then trimming and decreasing of some of these regions, that was happening less fast as a function of the stress on the child and the genetics susceptibility to ADHD. So you had almost inverse associations, but they were inverse because they were situated at a different part of the curve. One where you're expecting more increase, one where you're expecting a decrease. So that brings me to the part which is the part that I think is going to be the most key, and it has to do with how do we translate this into clinical applications? How do we go from these associations between prenatal stress, social environmental adversity, genetic susceptibility, predicts psychopathology 4 to 8 years later, even later. And it's really about, well, what are the pathways in between that we can then say are explaining that trajectory that then can be really the targets of early intervention and prevention. This is an interesting work done by the group in London of Argus Trankaras, which looked at 4,800 teenagers from the adolescent, the ABCD study. These are mostly European ancestry, and they examined what was you can see here, what were the mediators between genetic risk for depression and ADHD and psychopathology. So they knew that genes mattered in the prediction of psychopathology, and they wanted to know what were the pathways. BIS is a measure of temperament, and G is a measure of cognitive function, which includes, external executive functions and verbal and memory. And so it showed that you could find pathways between genetic susceptibility and mental disorders through examination of even temperament and cognitive function. They did so both using the prediction of the general factor or the individual sub-factors, which I talked about, externalizing, internalizing neurodevelopment. And they did so for both depression and for ADHD. That was very interesting because it's sort of like a proof of concept that you can start to find manners by which the developmental risk actually leads to outcomes that then you can maybe target. So I'm just going to develop right now, really, our model, our fundamental, sort of grand unified theory, if you say, for us, for our lab, that really guides the work that we're trying to do, which is we know that there's something called developmental susceptibility, whether it's prenatal stress from social environmental adversity, prenatal mood symptoms, genetic susceptibility, something that sort of looks like what the child is at birth. And we know that that is associated with outcomes of children in childhood, adolescence, and adulthood. Okay? What we need to understand is how does that happen? Because there's not a lot you can do, after the fact. You can certainly intervene with pregnancy and reduce stress and social environmental adversity, but that's a bit complicated, and genetics are certainly off limits. So it's about, well, what happens next if you know that there's a pathway, and it's really trying to understand the mechanism. And once you have the mechanisms, maybe then you can start to see how to intervene. So we think there are cognitive mechanisms, there's relational, like attachment, parent-child interactions, and they're also emotional, like negative emotionality, dysregulation. And I think I didn't do that explicitly, but when you think about some of the models of the eco-social model that Lawrence Kermeyer was presenting 2 weeks ago, and the work that he was doing with Ana Gomez Castillo, that these are some of the same sort of core psychological processes that really are instrumental. And the idea is that these are almost like a toolkit of what one encounters life stresses, which are sort of normal and expected in everybody's life, is what one negotiates these stressors with. And as a function of the ability of these abilities, whether it's cognitive, relational, or emotional, then that allows one to have positive or negative outcomes in face of adversity. But those are not ethereal. They didn't just emerge out of nowhere. We think they're actually on the pathway between developmental process, early developmental origins. And this then affects adolescence and then ultimately adulthood. That's really the model that we think makes, or assembles enough of the factors. And so major life stressors matter, all these processes matter, so does early development. And then we have to sort of look at, is it specifically internalizing, externalizing aspects of psychopathology that are predicted? Okay, so, and I'm just going to sort of dip into some of the work that our lab has done to show that there's reason to believe that. It's not yet replicated. That's the whole where we're at this stage, is now starting to harmonize these processes across all these cohorts. Okay. So for example, cognitive, that could be attention, it could be attribution, things happen good to me, things happen good to me only, when they're bad, otherwise when they happen, it's my fault. Otherwise, it's not my fault, it's by chance when it's good. And resilience, what do you do in face of like frustration? So I'm just going to talk about a couple studies. This is an interesting study that was done by Esther CK. She was finishing some of these associates that were run many years ago, and we looked at the prenatal adversity and genetic risk as a combined score of various different individual genetic candidate genes, for ADHD and maternal sensitivity, and showed that all three of those interacted to predict attention. So attention in toddlerhood, not like ADHD, just capacity to attention. So it just sort of gives you already one path. Resilience, that's something that I presented recently when we were doing our celebration of Leonora, who passed away last year, almost a year really to this date, and who had looked at hopefulness in 5-year-olds using a response to impossible puzzles. And just to remind, for those who don't know, you just give 5 puzzles. The first one and the last one are doable. The middle 3 are impossible. And then you ask the kid, how do you think you did? How do you think you'll do? And what do you think about the next puzzle? And it taps into sort of hopefulness and self-confidence. And you can actually break down children into 2 different groups. The kids who, after the first, puzzles, when the puzzles start to be hard, and then finally they have a good puzzle, they just still don't recover and they still don't think they've done well or they will do well. Whereas those kids who bounce back, because once they finally do a puzzle well, the last one, then they actually believe in themselves again. So you can sort of have 2 kinds of kid. And what's interesting is that you can predict that kind of resilience, hopefulness, from prenatal factors and from genetic stress factors. And this is what we saw, that genetically susceptible children who were exposed to low levels of maternal depression appear to be the group that is the most resilient overall. Whereas a combination of high genetic susceptibility and exposure to high levels of maternal depression were the least likely kids to exhibit overall resilience. So it just, even that sort of the resilience, sort of, I can do it. We have some kids, you look at them and they're sort of like looking at the puzzle and it's sort of like, they don't even bother. They say it's impossible. It's like, forget it. I'm not trying this. And then there's some who are literally throwing to pieces and, getting so upset. And when they start to do one properly at the end, they just say, it's okay. I'm just horrible. So it's always interesting too. And then so that's resilience, intention. And then emotional processes. What are interesting is, for example, there are 2 factors that have been identified a lot as temperamental traits called negative emotionality and dysregulation. This is the work of Vanessa Babineau, who's now in New York in our first R01, and did really wonderful work looking at the trajectory of dysregulation, and found that by 18 months, you have kids who sort of diverge in 2 different paths. You have the sort of dysregulated kids who at the age of 18 months start to sort of stay dysregulated, and the ones, which is about 90% of them, who actually are, regulated. So, and you can't actually distinguish before the age of 18 months which way you're going. There's a lot of factors intervening that actually modify that trajectory. But you can see that the ones who are dysregulated, after the age of 18 months, have a incredibly increased risk. So for ADHD, conduct, oppositional defiance, anxiety, depression, and comorbidity. It's almost an 8-fold increase in comorbidity that you could predict from this dysregulation trajectory. So we know that this dysregulation trajectory is meaningful to predict long-term psychopathology. But what we're trying to see is, can you predict the dysregulation from pregnancy and genetics? And we found that that was indeed the case. So you could actually find that a combination score of genetic susceptibility that includes process disorders, total problems, in interaction with maternal depressive symptoms, predicted dysregulation, which then predicted psychiatric comorbidity. So again, to tell you that there's a pathway, and then what can we do about high dysregulation? What can we do about that when we identify, knowing at the age of 18 months, it's sort of set, and then you can maybe do something about it before it leads to morbidity. Negative emotionality was the same. It's sort of fearfulness, worry, sadness, avoidance, and we can see that that even a couple of genes and interaction with maternal mood symptoms predicts this at 3 months, 6 months, 18 and 36 months differentially. So we know that there's an association between negative emotionality and genetic and prenatal stress. But what's interesting is, does this mediate the pathway to internalizing symptoms? And so what Catherine Gordon-Green did is she actually looked at a outcome of internalizing disorders and saw whether negative emotionality was in the pathway between prenatal stress and genetic susceptibility, and found also that association. This is the last paper for his PhD, which we're working on. And then finally, relational is how is, in our model, in this model, how are relational factors also mediating the pathway? Almost done. And this is really work of Shalaka Shah, who looked at the prediction of attachment and found the joint effect of both genetic susceptibility and prenatal stress in predicting attachment behavior, specifically avoidant. It's a very novel approach because instead of predicting specifically attachment categories, you look at the behaviors and adjust for the behaviors that exist in other categories, because all the three types of attachments, whether it's insecure, avoidant, or secure, have shared scores on various different axes, and typically it's treated as categorical, where it's actually it's rather dimensional, and then you just end up being scored as secure if you have more than the three other scales, not because you've been identified as being secure. It's just your behaviors are more frequent for secure, but you still have behaviors for insecure and for avoidant. So that was a very interesting study, because it showed that as prenatal stress increases, individuals with a high latent genetic score, meaning those kids who had more genetic risk for ADHD, psychiatric problems, sensitivity to the environment, the more prenatal stress experienced, the less avoidant behaviors they had at the age of 36 months. So it just showed, in a way, a sensitivity to the environment that modified later behaviors. So this is really gives you a sense of the kind of testing. It's one study, and really the issue is that one study does not make a finding large enough to lead to intervention. You need to replicate it, and that's what the consortium is really constructed around, harmonizing executive function, attribution, resilience, attachment, negative emotionality, dysregulation across all the cohorts, so that we can then test those pathways. What can that change going forward? Well, we can then do education intervention about the effect of prenatal adversity. We can even start to identify susceptibility profiles. We're doing that in a lot of machine learning models with the DOHAT group, to try and come up with an index of susceptibility that sort of upgrades the ACEs, because the ACEs is an index of adverse childhood event. It's got about like 8 or 9 items that you can add up from 0 to 8 and show an association with just about every adverse outcome, cancer and depression and heart disease. But what's interesting is it doesn't even include anything to do with pregnancy or genetics. So we think you can actually have a score of vulnerability or susceptibility that can be much more explanatory than the one that already is being quite touted as a very helpful approach. You can then start to think about which cognitive, relational, and psychological process you could invest in, especially if they're on the pathway. And then, and start influencing the early environment, which is some of the parenting. There's a lot of good work to show how that is modifiable. And what's interesting is how the postnatal environment really depends on developmental risk. So it's like we were hoping to identify who are the children that are more susceptible to these environments, because then, for better or for worse, which is the work that Belsky and Plus refined when they wrote their work on dandelions, tulips, and orchids, that there are some children, who are more affected by positive or negative environments than others, so that there's identifying who is more susceptible then might also then influence who you want to target for interventions, whether it's parenting interventions or other interventions around the cognitive and social emotional factors we talked about. And what's interesting is some children exposed to prenatal adversity, as we've seen with risk genes, might actually fare better when the environment later on is enriched. So the idea that you have everything is bred in the bone just because you have, genetic risk and prenatal risk, it really doesn't fare like that. It really also matters what happens subsequently. In summary, mental disorders such as depression and ADHD influenced by early developmental factors. Complex models are feasible and predictive. Harmonizing after the fact comparable studies is cost-effective for one, but also provides necessary replication to proceed to policy and intervention. After our papers, we don't have to say what's needed is replication. We can say, well, how big is this an explanation of the condition, and then what are the implications in terms of policy and intervention. And really, to do this, you're always looking for identifiable and modifiable pathways. Our future, we're harmonizing across all the studies. We're trying to develop, data that's not only at one level. So phenotypes, core constructs like, executive function and some of the tests that we do, and imaging and multiple environments. We're going to now we'll have to also include the role of major life stressors, which are, like we saw it happening throughout life and identified often in each of these studies around comparable time points. And I'm just going to put up again the name of all the people, including in the lab actively, who have been involved, all the collaborators in Maven from the past that have created this study that now we are reconsenting as adults to see how they're doing, and work of all the people in the executive originally. And like I've mentioned, all the different people across different sites. And if you have any questions, I'm going to leave. This is a very nice graphic. I really like, because if you can see the small print, each of these people are represented by a factor that are written across the body. It's a very nice sort of developmental sort of representation. And so you can toddler, ACE, 2, ADHD, 3, and it has all these different factors that are like emerging, and you can have them color coded. What are the infectious agents and ionizing radiation, tobacco, air pollution, alcohol, medication, environmental. Those are all the different. So it's a very nice representative of a DOHAD model that can cross this from an artist. Okay. Thank you.
Thank
Sure.
you. Who made the graphic?
Yes, who made the graphic? I am sorry, I don't remember. I'm gonna have to find out.
Okay, it's a beautiful one.
Isn't
I
it?
love yes.
I marvel each
Yes.
time I look at it. I marvel at the detail, and you can see all of this is all the black. What looks like black. These are actually all text.
Amazing.
Okay, so maybe I can stop sharing.
Yeah.
Thank you.
Yeah, thank you, Wazana. So we have time for questions. So we can see, you can enter them on chat or
Or
in
ask a
person.
question. Yes.
I can have a question.
Yes.
Thank you so much. I really enjoyed this. I wanted to know, in the studies that you mentioned they use the genetic susceptibility. Was it based on family history, the data that they collected, or did they actually do gene studies on all these participants?
Yeah, it's gene studies for all of them. Yeah. So that's why it's really hard to find comparable studies. because there are a few factors that are limiting. One is the use of genetics in the sampling of genetics in populations is often complicated. Starting in pregnancy is also very complicated. There are a lot of studies that have started later. Even like the big UK biobank has no information during pregnancy other than birth weight retrospectively, some of those studies. And the other limiting factor is because we care so much about the early environment is maternal environment, maternal-child interactions. And again, those are really complicated to capture. You need to have videos and code them. It takes years to capture that. But if you have that kind of detail, then you can really start to see what are the real players in the model. So that's left us with about 8 or 9 studies, I think, that are available.
Okay, Eric.
Valerie Ishak.
Yes, hi, beautiful presentation. I was just wondering how, if at all, any sort of paternal influence has come up throughout all of this, whether it be genetic or relational, or was it something that was controlled for at any point or is considered as a moderating factor, or has it come up?
Fathers, eh?
Yes.
So what we're talking about. Not that is unfamiliar from my personal experience, but it's many of these studies are from an era where there wasn't a lot of attention spent collecting information from dads. So I have to say that's the shortcoming of many of these studies. Even if I can speak from the perspective of the MAVEN, which was started only as, I think 2003. And even then, was quite blindsided by sort of fathered the collection. But when, 3-4 years ago, I said, okay, well, we should start collecting data from dads. Well, they weren't really very good at responding. And in part, we thought it was because we had sort of lost their buy-in because we spent so much time with the moms at the beginning in these experimental settings for attachment and for executive functioning and all that we didn't have really kind of so really I'm just speaking that even trying to repair that in the MAVEN was not very positive, and it is an important shortcoming of a lot of this literature, even from the genetic perspective, because we're always looking at the child's genes. And then sometimes we even have the mom, but there's like non-transmitted genetic information and the environmental effect that the non-transmitted genome can have on the environment that the child grows up in. So not only is it sometimes transmitted as a genetic susceptibility that the child actually has in its own genome, but some of that information is not transmitted, but actually modifies the kind of behaviors and the kind of environments that parents create for their children that we call genetic nurture. So it's really important to even capture that aspect of even the genes of the dad are very important, and we don't have that in many of the studies.
Okay, thank you.
Yes.
Thank you, fascinating. The concept of temperament and node very useful, and I'm wondering if if your scares it, this is sort of replaced by like a combination of cognitive, relational, emotional, and of course temperament is I'm not sure how you measure it because of the 9 parameters, but I always found it very much related to emotional regulation versus dysregulation, and very meaningful in terms of lifespan with Hayden's work and all that. I'm wondering what you think.
Just going to repeat the question because I think without mics, people don't hear, but the question of temperament and how it's an important factor that has been identified. Sometimes it's an 8 subscale measure. It didn't go into the details of the studies, but we use the Infant Behavior Questionnaire, which is the IBQ, and the Early Childhood Behavior Questionnaire, which is the CBQ, which are 2 measures designed by a number of sort of like foundational temperament researchers. when I have examined, we have examined negative emotionality and dysregulation, it is part of the factor structure that we created from all the subscales of the temperament measure. So they actually, some of them we actually created from our own data set because we were able to get them to concord at 3, 6, 8, 18 and 36 months. So for a rare occasion, we couldn't actually get them to converge in the same factor structure that originally Putnam had created. And then so we used the actual factor scores from the original study. But they actually are both negative emotionality and dysregulation are temperamental measures constructed using the subscales of these temperamental, classic temperamental. So yes, that's what, we actually see that that's a key piece. So maybe it wasn't clear, but that's what I meant by when we mean with social emotional is these kind of temperamental dimensions. And our docs now, really speaks to those as very important ways of categorizing person.
Eric.
Thanks so much. So I was, one of my questions was already asked by Valerie about how this whole literature is sort of flawed by the lack of attention to fathers. And we don't really know, it's sort of a black box almost, I guess, what the input of fathers might be on the outcome of these children. But anyway, I wanted to know as well, for inborn kinds of factors, whether genetic risk or temperamental factors that predispose to eventually developing a mental health problem. Can you quantify the mollifying effect of the environment, like in having a mother who's very attentive and appropriate, mature and resilient? Can you show effects of curbing the risk over time? Like, can you demonstrate that effect?
Yeah. So I forget the name of the investigator who did this, but Alsbach is one of the rare studies that actually captured father depression and mood during pregnancy. And his name will come back to me as soon as we finish. But originally he had produced a few studies that had shown that father's depression and mood symptoms during pregnancies as well contribute. There's actually a new textbook on paternal stress and the impact on child development. So that is a very rich field. But what was interesting is where originally he had identified that above and beyond maternal stress, father's stress and depression during pregnancy mattered. There was actually Rebecca Pearson, who's joined from the beginning with us, and then sort of tried to look at this study a little bit more. Did not replicate that. What we've seen though is that paternal depression postnatally is certainly much more significant. There's still some question about how the prenatal role of paternal. So I think that's a very important shortcoming that we have to acknowledge in our work. We have social supports, and and those are obviously linked in some ways with to cognitive, mental, emotional availability of both parents too. So quantifying the effect, we're not there yet. We want to be there. I'll have to tell you, there's one great study out of Pittsburgh that was done, a surrogacy study by Jenna Niederheier, and that was really the only unique study that's been done where they looked at the impact of the cohort is of these surrogacy pregnancies. So you have the donor, you have the surrogate parent, and then you have the adopted family, which really allows you to unpack the 3 different environments, which is, genetic susceptibility, because these kids whose donors, never really, involve in the virtual life, the surrogate who holds during the pregnancy, and then you can also measure stress during pregnancy and what experiences that has. And then sort of the postnatal environment, parenting, depression, coercive approaches for the expression of ADHD. It's a really very beautiful study. And what was interesting is it actually unpacks the attributional risk for each of those 3 and finds about 1/3 for each of them, so that you could try, I think they look at the expression of ADHD and even a condition that is thought to be so biologically driven. I think, you could say that there's a contribution during pregnancy and there's a contribution for kind of parenting approaches, maternal mood, depression, coercive parenting. So I think that's one of the rare studies that have tried to unpack that in as clean a model that can be. Ultimately, the idea is to try and see in the pathway how much of it can we explain by these various modifiable factors. Because we're really, and how much does the pathway even explain the variance in these pathologies? So if we're only explaining 10% of depression, even if 2/3 of it is explained by, I don't know, attribution, which, that's an important one, negative attribution, rumination, that might only explain 6% overall of the entire association. So our goal is really to reach something that's closer to, 20, 30% enough.
Yeah, I actually have a question. Do you think with the prediction model and the intervention sort of model that you're doing that there could be like a future where pregnant women would be flagged by their obstetrician or even children in maybe preschool or kindergarten for being at risk? And also, do you think that it can, in a way, it could also be potentially harmful?
Yes, that's a really big question. The group that I work with, which is called Towards Healthy Resilience in Varying Environments, Thrive, which is a subgroup of DOHAD that we created about 8 years ago, has exactly that mandate, was trying to come up with a susceptibility index with the explicit proviso that it's not unidirectional, but we're trying to talk about susceptibility, meaning not to say these kids are like bound to be, the very clear social impacting and operationalization of this research can be seen what France did under Sarkozy, where they just started to say these are kids that are going to be like that and really turn them into sort of negative sort of social exponent forever by saying these are the risks. And so we tried to run a longitudinal study in France about 15 years ago. And it was just impossible because everybody was worried of where this would be used. We ran also a sort of knowledge user group at the DOHAD meeting in Vancouver a couple of years ago, where we actually gathered a lot of people in this field, researchers and clinicians and policymakers, to advise us about this path from prediction to then application, and that was a lot of the conversation around that is what are the risks, what are the dangers, what are the safeguards, and it's fascinating if you just write what are the risks of prediction tools in psychopathology as in just an AI query. It literally comes up with like the top 10 items that this group came up with and the top 10 sort of remediation. So there are ways to mitigate the risk, but it is enormous for people to want to contribute information, not just about pregnancy, but even about their genetics. Knowing where's it going to go? Is the insurance company going to use it? Is the police department going to use it? Is it going to be used? So there's a lot. And then does this mean that, one, mental disorders are bad? Because there's certainly a portion of population that doesn't always see that sees that that's instrumentalized power through, medical profession. Two, does it mean that we are then putting the risk of mental disorders in the family when there's so many social and environmental factors? There's a lot of things that change, capital and neighborhoods and education and healthcare that, conceivably, if we increased wealth across the spectrum, we would be reducing most of the disorders. It's a fundamental cause. Socioeconomic status, position, education are fundamental causes, and they're not accidental. They don't belong only to people. They belong to so then there's the concern that we are too much centering the prevention in the people, in individuals, in families, and not in social organization. So that's a real concern, and then it's sort of navigating between all of that to say we are not deresponsibilizing, or we are not focalizing our, but we are saying, where can you actually do something? Where is there room to do? Where can you, if you have the chance to reduce the likelihood that your child's going to have depression at the age of 18 by doing X, Y, Z, it's the same with stroke and depression and heart attack prediction tools. You go online and you say how much you smoke, how much you drink, how much exercise you have, family history of depression, anxiety, of heart disease. And then it tells you in 5 years, 10 years, 15 years, this is the risk for all these things. Obviously, there are lots of social environmental factors that can affect, like neighborhood. In New York, depending on where you live, you might actually not have access to fresh fruit and vegetables. So there are certainly ecological factors that will influence. But if if I do this in 5 years or 10 years to reduce my risk of stroke or heart attack, would you want to do it? Social, environmental, unmodified structural factors aside. So this is not exclusion, and this is not just blaming, but it's just sort of, it's for us, we're trying to see, is there a way to empower people and just give them enough information that they can change some things in life that they can, if that on the life, we're saying conditions that have never changed in incidents. Never. I mean, it is absolutely unfathomable to think that there's still something about mental disorders that has been unswayed by anything we've done. But
Thank you.
yeah.
Thank you.
Any
questions online?
Oh, chat. Yes.
There's a few.
Sorry. Okay. Is there something I'm missing in the chat?
There said 3 there, but can't scroll up.
Oh, okay, there. Later. I know you're not able now. This is so interesting. Can we please speak in the future? Harriet, of course. Okay.
Okay.
I'm just typing. I mean, my email is very easy to find. It's Ashley Wazana at mcgill.ca. Please don't hesitate if you need to know more. Okay.
Yeah, I just had a question. So
Yes.
This one of your findings with neuroticism. First of all, is neuroticism and negative emotionality, are you referring to the same thing, or is that?
I guess neuroticism is a specific trait measured in adulthood.
Okay.
Versus negative emotionality is really a temperamental dimension, which it can be captured as early as 3 months.
Oh, okay.
And so it's like it captures like avoidance and fearfulness and loneliness and sadness.
Okay.
So, and it's like it's sort of a constructed from 8, or there's like about 9 subscales in the in the IBQ. I think has about probably 12 subscales, and the CBQ has another. So it's that's what it more or less captures.
Okay. And does it relate strongly to neuroticism?
I think so. I think so. I don't think it's we would call it sort of an endophenotype, just like we call neuroticism an endophenotype ultimately to a psychopathology, because it's not in of itself a disorder. There's sort of like a biologically related phenotype that's on the pathway, and probably biologically and transgenerationally influenced, but not systematically predicting a certain outcome. So that would be kind of an so it's a sort of a more mature version of the endophenotype of neuroticism, and yeah.
Okay. Then I know I was a bit confused by findings where neuroticism was negatively related to the P factor, but positively related to internalizing.
Yes. So it's interesting. So this is again the way that you sort of cut up the pie at that age, when you're trying to find what is shared of all the symptoms and what is specific and residual. So in a way, it's a model that says, okay, there's lots of symptoms that really sort of overlap. when you're looking at all the disorders in childhood, and that's sort of general. And you can actually see that it's a lot of the externalizing symptoms that sort of load on that. But then you still have a sort of specific externalizing factor, which is every symptom that didn't really load much on the general factor loaded on the specific externalizing. And then you have all the internalizing symptoms. So it sort of says neuroticism when in the face of, was it maternal mood symptoms? Yeah, doesn't predict general psychopathology, but it predicts more internalizing. So it's almost, it's you're less likely to have like more sort of over disruptions in behaviors and mood and all that, but more like internalizing. So it's not entirely counterintuitive because it's
Jordan,
so.
precursor, doesn't fulfill criteria. It's almost always, part of the traits.
Yeah. But the internalizing is closer to what we would consider to be depression, anxiety.
Sure.
That is the core symptoms that are left over that aren't just about, because, attention's affected, concentration's affected, sleep is affected. There's so many behaviors that are across the board, across most disorders. So you're really trying to see what is core left for internalizing disorders or externalizing disorder.
The generals, like maybe I misread the slide, but didn't it have anxiety on there, or is that something else?
Yeah, you load everything on it.
Yeah.
And it sort of takes whatever it finds that's common.
Okay.
And then what is residual is sort of parceled down to either internalizing or externalizing. You set up the bifactor structure so that the internalizing and externalizing are orthogonal. So they're in a way by design, they're supposed to not be overlapping.
Okay.
But the general factor is one that just captures what is shared as a variance. So and interesting, you can see over the ages what usually loads on that. Sometimes it's more internalizing symptoms or externalizing symptoms, but it has a very different sort of structure. Even as you go into adolescence, you start to add thought disorder, and into adulthood, you have sort of neurodevelopment. So it's very interesting that you can't even construct the same factor at every age group, because things start to be a lot more sort of separable as you get older. And you get domains like neurodevelopment and thought disorders and internalizing and externalizing.
Interesting. Any other questions?